Epigenetics is a phenomenon in which the genotype does not fully describe the phenotype. Paramutation is a subcategory of epigenetics in which a silent allele can silence a normal allele in trans in an epigenetic manner. This silent state can persist through generations even in the absence of the original paramutagenic allele. Currently, the mechanism of paramutation is poorly understood, and little is known of how paramutation shapes gene expression in natural populations. Here, we investigate a system of genic paramutation in Drosophila virilis that is mediated by piRNAs. In Drosophila, piRNAs act as an immune system to silence transposable elements. Transposable elements are selfish elements that can jump around the genome and cause DNA damage. Sometimes, however, piRNAs mistakenly silence genomic regions. Center divider, or CDI, is an example of this phenomenon. The left middle panel displays the cross scheme used in this experiment where a strain 160 female was crossed to strain 9 male, and the hybrid F1 females were then back-crossed to strain 9 males. Some of the first-generation back-crossed females will be homozygous for the CDI9 allele and lack the original CDI 160 allele. Previous studies by Irwin et al. Have shown that piRNA biogenesis from the original 160 allele is maintained across generations and may act in a paramutagenic manner. In this system of paramutation in Drosophila virilis, we believe the maternal piRNAs mediate CDI silencing. On the top right-hand side, titled piRNAs Map to CDI in Pure Strains, you can see that in germline and ovary tissues, piRNAs mapped only to strain 160. Underneath that panel, titled Expression of Center Divider is determined by a maternal effect, the data shows that the expression of CDI is lower in the germline of the F1 reciprocal progeny when the mother is strain 160, suggesting that the expression of CDI is determined by a maternal effect. This is further supported on the top right panel titled CDI piRNAs, reciprocal F1 progeny, where piRNAs map to CDI only when there is maternal piRNA present. Underneath that panel, the second-generation back-cross progeny have conserved piRNA biogenesis in the CDI over CDI9 females, even in the absence of the original paramutagenic 160 allele. This data shows that there is conserved piRNA biogenesis and repressed expression of CDI in progeny lacking the original 160 allele, thus suggesting that the expression of CDI is paramutagenic. The experimental approach is laid out in the bottom left-hand panel. The ovaries of 20 hybrid mothers and 20 CDI9 over CDI9 daughter pairs were extracted, and the RNA was isolated. Through an oligo-DT qPCR analysis, we measured the expression of CDI in the ovary tissue. 2 biological replicates and 3 technical replicates were conducted. The data was analyzed using a linear model in RStudio. The bottom right-hand panel displays the results. In this study, we found that the ovary expression of CDI9 over CDI9 daughters was significantly different than the ovary expression of the pure strain 9 controls. It is important to note that the CDI9 over CDI9 daughters are genotypically identical to the pure strain 9 controls at the CDI locus, thus characterizing this as a new system of paramutation. Further, when analyzing the genome assemblies of strain 9 and strain 160, we found that CDI is located at the telomeric region of chromosome 2. In Drosophila, transposable elements make up the telomeres. The dot plots at the bottom right-hand corner display 2 very different genomic structures at this telomeric region. In this region, we found multiple TART elements residing in strain 160 only. Future studies will focus on mapping piRNAs to this genomic region in strain 9 and strain 160 to better understand the nature of this system of paramutation.